Hello, Pavitra. Good to see you. You're the first speaker. Welcome.
Hi
Yeah,
there.
we can see you. Hi, Nayan. Good to see you too.
Hi, am I audible?
Yes, Nayan, you're
Okay,
audible. And
great,
Nayan,
great.
you have set a record. You're the only speaker who's using the background of
Oh,
our
okay.
background,
Okay,
and you
okay.
have revamped it up with a QR code also, Nayan.
Yeah, I mean, I just saw that. Okay, yeah, thanks.
So,
Yeah.
Nayan, brownie points plus points for that. I'm taking a
Okay,
screenshot.
sure. Okay, yeah.
Super well done. Thank you. Thank
Okay,
you. Hi,
welcome,
Deepa.
welcome.
Good morning.
Yeah.
Hi, hi. Thanks for joining, Deepa. And yes, who are we waiting for? Silpa and okay. Pavitra, your slides are ready?
Yeah, they're ready. Sorry.
Okay, okay, okay. Great. We are also live on YouTube. Yeah, we'll start in a minute. We are waiting for Viraj also to join. So all the best, Pavitra and Nayan. Yeah.
Thank you.
And I will say in my introduction that we actually got the least applications from ecology and evolutionary biology.
Yeah.
I think everyone is in your area of work, everyone is doing field work. Nobody has time for webinars, possibly. But we received the least applications actually. Pata nahi kyun. Maybe we can publicize it more. Okay. So I think Viraj is also joining. Na? I think I saw him. Great. Great. Let me start. Screen sharing at least. So that's great. Really, super, super welcome to you all. Wonderful to find you here on a Friday at 10 a.m. Welcome to PhD Cafe. It's been going really well through the week and just waiting for Viraj to join. I can see he's connecting to audio. But yeah, I think you're there, right, Viraj? Yes. So it's been a super exciting week. Welcome to our speakers, Pavitra and Nayan. Welcome to our audience. Really great to see you all. That's some commitment. Some of you have been with us through the week. That's some super commitment from you all to listen to all the talks. And welcome to our expert mentors, Deepa and Viraj. Thank you for your time. So this is the lineup for today. Today's session is ecology and evolutionary biology. As I was saying, very happy to be hosting this topic. We didn't receive too many applications from this area of work. Something for us to keep in mind. But thanks, Pavitra and Nayan, for your interest. And we're looking forward to your exciting talks. General instructions, of course, hear us, see the slides. Keep the chat window active with your questions, suggestions, feedback for the speakers. Each speaker will be given 20 minutes, Pavitra and Nayan, and we are sticking to time. Okay, so 20 minutes and then 10 minutes for expert mentor Q&A. That's from Viraj and Deepa. And if we have time after that, we may open it up to audience questions. But the audience will keep typing questions in the chat window. So even after your talk, you can address them in the chat window. And of course, feedback should be constructive and to better the work and to advance the work. All right. So our mentors for today are Viraj and Deepa. Thanks for coming on board with just an email. You all have a lot going on, and we appreciate you giving this time to the community. Thank you. Um, and the audience. There's a lot of good science to be discussed, but there's also an opportunity to win a physical copy of the India Bioscience ebook put together by my colleague Ankita. And it's getting past the finish line, and we've done a play on the word finish. So this physical copy can be won by you. What you need to do is keep the chat window active. And we have given eight giveaways so far. Today is the ninth giveaway. So you can get the ninth copy of the book. So keep the chat window active for that. All right. With your questions, suggestions, feedback, and so on. Okay. So hope that's clear. And this session will end early, but at 1 p.m., we have an open networking session, another meeting, another link, and that will be hosted by Arushi and Ankita. And Arushi will be telling you about transferable skills to develop during a PhD, and Ankita will be talking to her about it. And just to tell you, my colleague Arushi at India Bioscience did a PhD in genomics, and she's very successfully transitioned to her role as program manager digital initiatives, where she hosts webinars, podcasts, writes articles, facilitates meetings, and so much more. So, how did this very quick transition happen? And what were the skills that helped Arushi make this transition? We'll talk about it shortly. All right. So, with that, I think let's get started. Pavitra, share your slide.
Sure. Can you see my screen? Okay. Okay. Good morning, everyone. My name is Pavitra. I am a fifth year PhD student. I work with Professor Supreet Saini in the Department of Chemical Engineering in IIT Bombay. Today, I'm going to be talking about the mechanisms and consequences of adaptation of a non-functional horizontally transferred gene. Now, for those of you who are new to this, this whole area, I'd like to give a small bit of a background. So let's just take a step back and ask what are the different ways of doing a task. And let's say that task is that I have to write an assignment, right? So the first and the most obvious way is that I do it myself. It's obviously sad and boring, but more importantly, it takes time, it takes effort to fine tune it, and maybe I just cannot do it before the deadline that there is. Alternatively, I can borrow a copy of the assignment that my friend just did. And this is very easy. It's quick. There's really no effort, and I get a ready-made solution to my problem, very quickly. Much like how we exchange assignments, living organisms also exchange genes, and this is called horizontal gene transfer. Now, horizontal gene transfer is an extremely important evolutionary mechanism that has shaped life on Earth. And until HGT was discovered, we thought that evolution could be represented as some kind of a tree of life. But after HGT was discovered, we started seeing these, connections between the branches of the tree. And now we look at evolution as some kind of a web of life. But not all horizontal gene transfer events are successful. They depend on a bunch of factors. And I'm just going to list those factors. The first is that where is this gene getting inserted in the new host genome? Is it getting inserted at the right spot? Is there a good enough promoter in front of it and things like that? The second is the gene getting effectively transcribed and translated? And this is extremely important for us to get a functional protein. And is the mRNA and protein that's produced as a result of transcription and translation stable enough? And is the protein that's produced being trafficked appropriately to the right spot in the cell? And lastly, and most importantly, can this protein confer some kind of novel functionality to the host cell that it is in? Because if this horizontally transferred gene is going to code for a protein which performs a function that the cell already can, there is no reason for the cell to keep this foreign gene and make the proteins for no reason. So essentially, we need this horizontally transferred gene to confer some kind of a novel functionality. Now, in a scenario where the gene is not functional, we ask how it adapts. And we do that by using yeast as a model organism. So yeast has three ammonia transporter genes. So these genes code for proteins which get embedded in the membrane and bring in ammonia from outside the cell to the inside. Now, cells which do not have these three ammonia transporter genes cannot grow in low ammonia environments. Now, we create synthetically these yeast strains, which, by knocking out MEP1, MEP2, and MEP3, and we ask if ammonia transporters from other organisms can make these yeast cells grow. Right. And from literature, we see that many ammonia transporters, namely from E. coli, C. elegans, Arabidopsis, humans, can restore growth of this triple mutant yeast, whereas the ammonia transporter AMTA from Dictyostelium cannot. And Dictyostelium is an amoeba, it's a social amoeba, and we see that AMTA is one of the ammonia transporters in Dictyostelium, and that cannot restore growth in triple mutant yeast. We verify that by transforming our triple mutant yeast with AMTA, and by growing it in low ammonia conditions. And what you see on the right side of your image here is the wild type yeast cells, which have MEP1, MEP2, and MEP3. So clearly there is, the cells which have MEP1, MEP2, and MEP3 can grow, but the ones which have AMTA cannot. Why is this the case? And it is a little surprising because all of these ammonia transporters came, they're said to have a same, they're said to have a common origin, and they've diverged at different points in evolutionary time scale. And and we just saw that ammonia transporters from E. coli and C. elegans and other organisms can confer growth in yeast. So this is also surprising from the context of the sequence similarity, because if you take any two ammonia transporter genes, their similarity is between 15 and 92%, but most of them are about 60 to 70% similar. So we ask what is wrong with AMTA in yeast by fusing it with GFP and performing confocal microscopy. And as a control, we also fuse MEP2 with GFP. We know MEP2 is one of yeast's ammonia transporters, and it's functional. So we fuse MEP2 with GFP, AMTA with GFP, and we perform confocal microscopy. So since MEP2 is functional, the image that we see should be something like this, where there is clear fluorescence on the plasma membrane. But in case of AMTA, we don't know what the problem is. So if the problem is that there is not enough protein that's being produced, then we would see something like this, where whatever is being produced is getting embedded in the plasma membrane, but it's not enough. The other possibility is that the proteins being produced in sufficient quantities, but then it does not localize on the plasma. So then we'd see fluorescence in the entire cell. And our results indicate the second possibility, that AMTA suffers from a localization problem in yeast. So the image on your left clearly shows that MEP2 fuses, MEP2 localizes clearly on the plasma membrane, but that's not the case for AMTA. We see fluorescence throughout the cell, and it appears like it localizes on some kind of an intracellular organelle, and we're not sure what that is. So we go back and ask where AMTA localizes in Dictyostelium, which is its native organism itself. And we found this study, which is about, a little over 15 years old, where they show that AMTA localizes on these two intracellular organelles, in endolysosomes and phagosomes. And its function is to essentially export ammonia during the life cycle of the amoeba. So with this information, we ask what is the minimum number of mutations that are required to make AMTA functional in yeast. And what we mean by this is that AMTA's function should flip from exporting ammonia to importing ammonia. And we focus on this minimum bit because if this gene, let's say, needs 10 mutations to become functional in yeast, it's likely going to be thrown out by the cell by then, because acquiring 10 mutations is not easy, and the likelihood of retention of AMTA in an evolving yeast population is going to be really low. So we want to ask what is, in some sense, the probability with which AMTA will be retained in this triple mutant yeast. And secondly, we want to know if the beneficial mutations are localized to any part of the gene. And we ask this question because from cell biology, we know that the signal peptide is important to decide where the protein goes in the cell. Now, for membrane proteins like AMTA, the signal peptide is essentially the first transmembrane channel itself. So we want to ask if these mutations are localized to the first transmembrane channel, or there is any pattern at all where these beneficial mutations are. So we do this using directed evolution approach. So we created this mutant library of AMTA. These are random mutations which, in each allele, and there are between 1 and 5 mutations in each allele, and we introduce them to, triple mutant yeast and see which of them can confer growth at 500 micromolar concentration of ammonia. So what we do is that we plate them in 500 micromolar plates, and we screen 500 alleles, and we got 9 functional ones. And this is what a typical low ammonia plate looks like, where you have a yeast cell, where you have yeast cells with a functional allele of AMTA. Now we got these alleles sequenced, and also we waited up to 10 days of streaking on low ammonia plate. So these alleles, these 9 alleles came up between 4 and 10 days of streaking. So that indicated that there are, many ways by which AMTA can become functional in this new environment. We got these alleles sequenced, and surprisingly, there was clearly no localization of these beneficial mutations. They were all over the place. This gene is about 450 amino acids long. These mutations were spread throughout the gene, and there was also no pattern in which the amino acids changed. And interestingly, we also found this one synonymous mutation, which was able to, confer growth in yeast, although very low for fitness, but it was able to confer growth in yeast. Now, with this, we wanted to make sure that these alleles, functional alleles, can also solve. They're restoring functionality by solving the localization problem. So we did the whole GFP fusing and confocal microscopy again for all the 9 alleles. I'm just showing you 5 here, and for all 5 of them, we see that there is a clear change in the localization pattern. And it's extremely surprising that SNPs and in one case even even a synonymous mutation can do this. Now, with this, we know that AMTA can very easily become functional in yeast. It's also very quick because it just needs one mutation, and there is really no constraint. There appears to be no constraint on where these mutations should lie. We isolated 9 variants, and I'm sure there are many more. So with this information, we want to ask 2 questions. One is that, are there any byproduct and collateral effects? So what I mean by byproduct effects is that, remember that we isolated these alleles based on their ability to confer growth at 500 micromolar ammonia. We want to ask if by virtue of adapting to this selection, they've also gotten some other ability. And what I mean by collateral effects is that, do they have this some other ability which could also be harmful to the cell? Right? And the second question that we ask is, what does the landscape of AMTA look like? And I'll come to that when we get there. So we grow these yeast cells carrying these different AMTA alleles in batch cultures in 2 different concentrations of ammonia, 500 and 5 micromolar. And what you're seeing on the y-axis is a measure of fitness. We use growth rate as a measure of fitness, and that is normalized to the wild type. And we see that most of these AMTA alleles have environment-dependent effects. Although they were isolated at 500 micromolar, all of them do better than the wild-type yeast cells at 5 micromolar. And this is surprising because yeast, so I told you yeast has 3 ammonia transporters, right? MEP1, MEP2, and MEP3. The 3 of them have different sensitivities to ammonia. So MEP2 is the most sensitive one, which can import ammonia at even when its concentration is very low. But MEP1 and MEP3 are not as good. So despite our selection not being for import at low ammonia concentrations, all of these AMTA alleles do better than the wild type when it comes to lower ammonia concentrations. AMTA3 alone is the only allele which does not show environment dependent fitness effects. Now moving on to the collateral effects. So the name MEP comes from the fact that these genes can also import methylamine from the environment. So MEP stands for methylamine permease. And methylamine is toxic to the yeast cells. So cells which have this MEP1, MEP2, and MEP3 cannot grow in methylamine environments. And we want to ask if any of these AMTA alleles can also import this toxin and end up, killing the yeast cell. So again, what we do is we grow these yeast cells with these alleles in batch cultures in 2 different concentrations of methylamine, 100 and 200 millimolar. And what you're seeing here is again fitness. We use growth rate as a measure of fitness, and we see that the wild type, which has MEP1, MEP2, and MEP3, has really low fitness. But this triple mutant with AMTA is doing much better than the wild type, and that is also the case for most of these alleles whose fitness is way higher than the wild type in both the concentrations of methylamine, except this AMTA6, which has a really low fitness at 200 millimolar, indicating that this is importing methylamine from the environment. But interestingly, this import is contingent on the concentration of methylamine. So that is something that is very surprising that we saw. Now moving on to the landscape bit. So what do we mean by a fitness landscape? So a fitness landscape is essentially this. When I plot fitness against genotype and I ask what this graph looks like, that is what fitness landscape is. And let's say I have a landscape like this, which has just one peak, and let's say I have 3 different genotypes: green, gray, yellow, and blue. If these populations are allowed to evolve, they will all eventually go to this single peak, and by virtue of natural selection, they cannot decrease their fitness. So natural selection does not allow any mutation which decreases your fitness. Right? So as these three genotypes evolve, they would go to the single peak. But on the other hand, let's say I have a landscape like this, which has three peaks and two valleys. Here, what decides the final fitness that a population would reach depends on where it is starting from. So let's say I have these three genotypes, this yellow, gray, and blue. This yellow one, because it is where it is, it can only access this lowest fitness peak. And again, like I told you, natural selection does not allow downward movement, so it cannot access any of these other two peaks. But the gray one, on the other hand, can access one of these two because it appears to be somewhere in between these two peaks. But the blue one is kind of stuck because any movement from this peak would just mean that its fitness is to decrease, and it cannot reach a higher fitness peak. So this blue one really cannot move from this peak to the higher one. So we want to ask, so these two landscapes are called smooth and rugged landscapes, and we want to ask what AMTAs looks like. And one way to ask that is by measuring the number of beneficial, deleterious, and neutral mutations available to each genotype. And what I mean by that is the following. So let's say I take this yellow genotype on the, on my right landscape. This one can improve its fitness in a few ways, but it can also decrease its fitness in a few ways by, undergoing deleterious changes. The gray one, on the other hand, has a really low fitness now. So there are only, there has to be many mutations which only improve its fitness. But the blue one is at a fitness peak, and it's most likely going to have only deleterious mutations because there's really no way that it can improve its fitness from where it is. So we want to ask what AMTA's landscape looks like, and we do that by picking two AMTA alleles, AMTA1 and 2, and we generate like 100 single mutants of each of these. So this is AMTA1, it has this yellow mutation. We generate 100 mutants of each of these, of AMTA1 and AMTA2. Now these are random mutations. We do not we do not know where they occur, but all we know is that they are just one mutation different from AMT1 and AMT2. uh, Let me just remind you, AMT1 has a greater fitness than AMT2. So then we put all these 100 mutants back in our triple mutant yeast cells and measure their growth rates. And then we compare the growth rate of this, one of these 100 mutants with their immediate ancestor. So let's say the fitness of AMTA1 was X and the fitness of this, the first allele was Y. We ask, is Y greater than X? Is it lesser than X? Is it the same? And depending on that, we classify them as beneficial, deleterious, and neutral. So we measure the number of beneficial, neutral, and deleterious alleles for each of these AMTA mutants and ask, what the shape of the landscape would look like. So this is what it looks like for AMTA1. Most of the mutations are deleterious. There are very few ways by which it can improve its fitness. And how you read this landscape is let's say I pick this bar which corresponds to minus 0.6. That means that there are about 8% of mutations which have a fitness effect of minus 0.6. The graph looks almost very similar for AMTA2. In fact, it has more deleterious mutations than AMTA1, but again, the opportunities for fitness gains are very limited. And what this indicates is that the landscape of AMTA is quite rugged. There are many fitness peaks, and our functional AMTA alleles seem to be stuck at these fitness peaks with like very few opportunities to improve their fitness. Yeah, that brings me to the end of my talk. I want to summarize the most important findings, and I think that has to do with the fact that there are many non-localized SNPs and a synonymous mutation which can independently solve the protein localization problem. We do not know how we do not know the mechanistic cell biology basis of why these mutations are able to solve this localization problem, but this is a very surprising finding. And they do that by flipping the membrane affinity of the protein. So the wild type AMTA localizes on intracellular organelles which have a convex surface, but these mutations make AMTA localize on the plasma membrane which has a concave surface. And by doing this, by solving this protein localization problem, it aids protein evolution and neofunctionalization. Remember that this AMTA was not able to import ammonia into the cell, but now it does. And we also saw that it can also, one of these alleles can also import methanoid. And in the context of this work, they make horizontal gene transfer successful. And what this indicates is that if AMTA were to move from dictyostelium to yeast, it's very likely that it's going to be retained because there are many ways by which it can become functional. And when we started out this work, we had our baseline expectations that we should have like at least localized mutations, 4 or 5 of them, which make which change how it localizes. But at the end of this work, I'm only reminded of this quote from one of my favorite movies, which says sometimes it is the people no one imagines anything of who do things that no one can imagine. I just want to replace people with SNPs because we really did not expect that SNPs could just change the whole way AMT worked. With that, I want to thank my advisor, Supreet, for the opportunity to work on this fun problem. I also want to thank Sudipta and Nitika, who are my lab mates who helped me with this project. I also had the opportunity to spend a year in Sergey's lab at UC San Diego. I'm very grateful for the inputs and feedback that I received from my lab mates there. I'm funded by IIT Bombay and PMRF. My stint in the US was sponsored by Fulbright. I want to thank India Bioscience for the opportunity to talk about this work. And thanks to all of you for listening and showing.
Thank you, Pavitra. Great
Yeah,
talk.
thanks. Thank you.
Well within time. Well within
Thank
time.
you.
So we have a full 11 minutes for Q&A. We'll start with Viraj and Deepa and then audience Q&A if we have time. But there's some questions for you in the chat window. Okay, you
Okay.
can deal with them. Go ahead, Deepa, Viraj.
Okay.
Okay, very nice talk, Pavitra. Maybe I'll go ahead and ask a couple questions.
Sure.
One, I was wondering, have you quantified the differences in the distribution of fitness effects of your AMT1 and 2 alleles? They looked similar, but maybe it's worth quantifying them to ask whether there are any differences, because you do expect there should be differences, right? Given that AMT2 has a lower fitness than 1, or other way around, I forgot which way.
AMT2 is lower, yeah.
Okay, so have you tried?
We want to do this for the other 3, single nucleotide alleles as well. So then we want to draw a complete kind of observation of what is happening with the DFE. So we haven't done that yet.
I see. Okay. And are you going to include the synonymous beneficial allele as well as one of your
Yes.
backgrounds?
Yes.
Oh, that'd be wonderful. Okay, great.
Yeah.
One more question I had was, I was a bit confused. You said that the membrane affinity is flipped from concave to convex. Could you explain to me what you mean by that, and what do you mean the membrane is concave versus convex?
So the wild type AMTA localizes on intracellular organelles, right? And their membrane is like this, and AMTA localizes here. But plasma membrane is like this, and AMT is going this way. That is what I meant.
I see. And you
Yeah.
think the concave versus convex nature or whatever, wherever they're sitting, has anything to do with that localization, or?
Because so I think from the images that we got and from the study that was conducted, with dictyostelium itself, so AMTF localizes mostly on the intracellular organelles. But after these mutations occur, it goes straight up to the plasma membrane. It doesn't localize on any of the intracellular organelles. So I'm guessing it would have something to do with that.
But it's functioning to move ammonia like you'd expect it to, even when it is in the inner membranes, right?
That's true. Yeah. I think it's still sensitive to higher ammonia. It's still pumping in ammonia from our region of higher concentration to lower, but it is but the shape, or I think the shape of the membrane on which it's localizing is getting changed because of these mutations.
I get that, but I don't see how that has anything to do with the function. You see what I'm saying?
Sure. Yeah. That's
It's
true.
still moving
it's
ammonia,
still,
right?
Yeah. From a region of higher to lower concentration. Yeah.
Okay. And last question. Do you know, is it possible at all to put your modified AMT alleles back into dicty and ask what happens there?
I'd love to do that, but I'm not sure I
Sure.
have the expertise to do it.
Okay. All right. Fine. Thanks.
Thank you.
Hi, Pavitra. I really enjoyed your talk. This
Thank you.
is not a field that I'm an expert in, but I learned a lot from your talk. So thank you.
Thank you.
I especially liked how you gave a broad overview initially. I just thought that you could have spent a little more time, maybe having a slide dedicated to talking about the broad and the specific questions that you have. I thought that part was a little missing.
Okay.
Also, throughout your talk, I liked how you took time to pause and explain certain things. Like the fitness landscape was extremely well explained.
Thank you.
Um, but the part about there were some parts like the methylamine import, and when you compared AMT and MEP2, it wasn't clear to me why that was done. Yeah, so that part I thought could have been maybe explained a little better.
Okay. I can explain it to you if that's fine.
Sure. My reason to mention this was potentially to improve the presentation. If you
Of course.
agree, it could be included.
Of
But
course.
I would love that explanation. Maybe can I make another couple of points before
Of
you?
course. Yeah, sure.
I was also thinking whether you have wondered, and this is not something you have done, but I wonder how for horizontal gene transfer to happen, of course, these cells have to interact, right? So I wonder how the ecology of these species links into what you have done. For example, do Dictyostelium discoideum and yeast encounter each other often in the wild?
No, there's not a lot of evidence of them coexisting in the wild.
Yeah, and I wonder, broadly speaking, how likely or unlikely HGT would be happening between two different species given their ecology.
Yeah, so HGT is obviously most likely between organisms which are living, with close to each other when there is physical proximity. But I think the focus of this work was not to, like stick. We just used AMTA and yeast as a model system, and we hope that we can see similar results with other model systems as well.
I agree, but I think what you've done might be actually getting onto something even more interesting because, for example, convergent evolution happens um, more often than is expected, sometimes, at least in some groups. And something like a GT could really explain it well, even though species might be very far from each other. If you do have this chance event of encounters happening, and if it does if a GT does lead to like a functional um, boost of some kind, it could really explain some of these patterns that are relatively poorly understood.
Sure, yeah. And another kind of thing to note is that viruses are very popular carriers of, genes from different organisms. I mean, that's not relevant to yeast and dicty, but in an ecological scale, viruses keep shifting genes from organisms. So even if they're really not physically close to each other, there is this possibility that they could get genes from other like far away organisms as well. So yeah, I think what you're saying is worth thinking about. Yeah.
Sure, that's a good point.
Yeah.
Thank you.
Thank you. Yeah.
Thank you, Deepa. Thanks, Viraj. Well done, Pavitra. Great
Thank
talk.
you.
It sparked
Yeah.
a lot of good questions. Pavitra,
Thank
you
you.
want to take one or two from the chat window since we have time till 10:35?
Yeah.
That's it. Dibyo asks, what's the efficiency of horizontal gene transfer in heterologous organisms? Are these mutations spontaneous or induced?
Efficiency of HET. Okay. I don't think I have an answer to that question. The mutations, so if you mean the mutations in our study, are they spontaneous or induced? In nature, they're, I mean, if AMTA moved from Dictyostelium to yeast in nature, the mutations would be spontaneous. But here in our study, we create this artificial library of AMTA mutants. So they are not, I mean, we don't let these yeast cells evolve. We create this library and we introduce them to yeast. So that I hope that answers the Bill's question.
Sure. If you want to elaborate, you can do it in the chat window. And there are a couple of more questions also.
Okay,
You
I'll
can
let.
take them. All right, Pavitra. We'll move on. You can
All
answer
right.
them in the chat window.
Of course, yeah. Thank you.
Thank you. All
Yeah.
the best. Best
Thank
wishes.
you.
Well done. All right. Next speaker is Nayan. Hi, Nayan. Nice to see you.
Hi,
And
hello.
all the best.
Oh,
And yeah, I really like your background.
oh, thanks. Yeah.
Yeah.
Thank,
Good. Go ahead. Slide share. You want to
yeah,
share your slides? Yeah.
yeah, I'll share this.
So, Nayan, you have 20 minutes.
Okay, yeah.
Yeah.
Lights are visible, right? Uh,
Yes, they are visible. They're
Okay.
visible.
Okay. Hello, everyone. Good morning. Thanks for joining. And thanks to India Bioscience for providing this opportunity. So I work in a slightly different field. While I applied to India Bioscience, I applied under other. So I work in origins of life here. And yeah, so India Bioscience put me in the most conducive uh, categorization. So today I'll be talking about dynamics of compartmentalized self-reproducing ribosomes. And let's get started. So just to give a broad context to origins of life. So on the right-hand side, you see the extant form of life, which goes back to Luca, and these are all the kinds of creatures that you know. And on the left-hand side, we see habitable worlds, and the only habitable world currently we know is Earth, though that's in progress. And in between, there are many steps through which one can go from a habitable world to uh, diversity of life, and especially through the first steps is prebiotic synthesis, where small monomers are produced from the prebiotic environment in Earth. And then the second step is when these monomers can combine to form certain polymers with functions. And the third step is some of these polymers could be self-reproducing systems, and they can be compartmentalized into what is known as a protocell, which is like a precursor of a cell. And then these protocells can evolve towards greater complexity to give rise to known living forms. And broadly, there are two kinds of approaches in the field. One is a top-down approach in which one starts from extant organisms and takes certain RNA or DNA and then tries to reconstruct some of the steps in between. Or one can start from a bottom-up approach in which one can start from simple chemicals and precursors and then find the conditions under which self-reproducing systems and compartments can emerge out. So let me give a review of some of the self-reproducing systems that have been studied. So there are broadly three different categories here. So the leftmost is a DNA-based system. It was first reported by von Kiedrowski, where you have a 6-nucleotide oligomer, which acts as a template. And if one starts with certain triphosphates, 3 nucleotides each, the 6-nucleotide oligomer can act as a template to bind these triphosphates, which can then facilitate a covalent linkage between them and form the template itself, and this can go in a cycle. The same goes for this protein-like system, which is engineered from a transcription factor of yeast, where one can start with the fragments, which are small peptides, and then start with the template, which is the protein molecule, or it's actually peptide, so it's around 30 amino acids, and this also can template the monomers, and the monomers can again link to form the peptide itself, and this can go in a cycle. Similarly, there are the RNA-based system where one can start with RNA fragments, and these RNA fragments can self-assemble to form a functional RNA molecule, which is called a ribosome. And ribosome is an RNA which has a catalytic activity also. And this ribosome can then autocatalytically produce more copies of itself. So for my study, we chose an RNA molecule, partly because of the RNA world hypothesis that proteins don't have information heredity, and DNA does not have catalytic function in the traditional sense. And with the discovery of ribosomes, this kind of became a bridge because ribosomes can have both uh, genetic information and catalytic activity. So let me dive into it. So the talk will broadly divide into two major parts, where I first go into the self-reproducing ribosome system that we studied and the compartmentalization strategy that we employed. And the second part is when you do certain environmental driving. In our case, it's temperature, and then you show very crude signatures of selection. So the self-reproducing ribosome system that we work with is derived from Azoarchus group 1 intron, and it's a self-splicing ribosome. It has been engineered in vitro to spontaneously self-assemble. So this is the secondary structure of the ribosome. So if this can be fragmented into oligonucleotides of like 20, 30 nucleotides in length. So basically 4 fragments, 5 fragments, or even up to 7 fragments. It can be fragmented into. And these fragments, under certain conditions like temperature and cofactor concentrations like magnesium, they can spontaneously self-assemble to the non-covalent version of the ribosome. And many of these non-covalent versions of the ribosome can template each other to form the covalent version. In that sense, it's autocatalytic. Even the covalent version can also template and form more copies of itself. The mechanism through which it really works is something called internal guide sequence, IGS, and tag, through which, so basically 3 nucleotides in the 5' end and 3 nucleotides in the 3' end. These are called IGS and tag, which turns out to be the major recognition sites for it to template and then autocatalytically form more copies of itself. One has also shown that if one does mutations in these critical regions called IGS and tag, one can create networks of RNA in such a way that one ribosome catalyzes another ribosome, and the second ribosome catalyzes the third one, and the third one can catalyze the first one. So one can form certain networks of ribosomes which can then collectively self-reproduce. So one can either have single ribosomes which can catalyze its own formation, or one can have like networks of ribosomes which can collectively catalyze. The way it works is based on the recognition, which is mostly Watson-Crick pairing. So in that sense, and some Abel pairing is also there. And so this is the ribosome system. And for the compartment system, we wanted to study quaservates. Quaservates have been hypothesized in the original life field since the beginning by Oparin and Halden. So these are membrane-less compartments which are very low in complexity, that don't require lipid vesicle or a membrane to be there. So how it really forms is one can take certain simple molecules like polyacrylic acid and spermine, and these are negatively and positively charged molecules. And what happens is, under electroneutrality conditions, when the charge is equal, they phase separate out to form these compartments. And initially, we optimized the concentration of this, the positive charge molecule and the negative charge molecule, and we found that, okay, there is a certain optimum concentration in which they form the maximum quaservates. And here is an image of quaservates here. This is in bright field, and this is under fluorescence. And this is basically to show that most of the fluorescence, which is because of a negatively charged oligonucleotide, is concentrated in the volume of the molecule itself. So it kind of concentrates molecules in the quaservates. So with this strategy to form quaservates, we then wanted to integrate RNA into the quaservates. So we went ahead with the positive and the negative charge molecules and RNA. RNA is also negatively charged, and we tagged the RNA with Alexa, which is green, which gives fluorescence in the green channel. And then we developed this protocol in which, so we first mix all these constituents in the RNA fragments along with the positive and negative charge molecules in certain ratios, and then either we centrifuge to get a thick pellet and incubate this at a certain temperature, or we just incubate it, in which we have the droplets and not a thick pellet. These are the 2 different phases in which this quaservate can exist. One is this thick pellet, which is very large. We call it quaservate macrophase, and the other is the small micron-sized droplets. And so this is what we call quaservate droplets. Both of them, as you can see, are green in color. So what that means is the RNA that was tagged with the fluorophore is mostly concentrated in this quaservate phase. And then later on, we incubate it at certain temperature for certain time, and then break these quaservates by adding extra charges, and then run it on a polyacrylamide gel to analyze how much RNA has actually reacted, how much fragment of RNA has actually reacted to form the RNA product. So to show, left side is a schematic where one has the polymers and the RNA molecule. They form these quaservate droplets or the quaservate macrophase. This is the image of that. And then on the right hand side, these are gel images, and this is a time component. This is up to 6 hours, and these are gel images. And what you see is defined fragments. So these fragments are called, so they have names like W, X, Y, and Z. So we start with a 4-fragment system, and then they can form, they can combine in an autocatalytic manner to form a 2-fragment, 3-fragment, and a 4-fragment ribosome. So the entire ribosome is called WXYZ. And so this is what we, from the gel analysis as a function of time, this is what we plot And these are three different systems. So one is the coacervate macrophage, which is this one. The other is the coacervate droplets, which is this one. The other we call bulk, which is basically the just ribozyme reaction in liquid phase, non-compartmentalized aqueous phase. So, and what you see the trend is both in droplets and macrophage. So basically in condensed phase or in compartments, the fraction of the intact ribozyme, which is WXYZ, the complete ribosome, which is this band here, the fraction of this ribosome in all three increases with time, but the rate seems to be higher in both the droplets and the macrophage as compared to bulk. So compartments kind of offer a certain uh, advantage to assemble ribozymes at a faster rate as compared to just aqueous phases. And to show the autocatalytic manner of this self-assembly, which is as a function of, so if we dope these droplets as a function of intact catalyst, which is the WXYZ ribozyme, and as a function of this catalyst, there is a clear linear relationship with the initial rate. So this is kind of a signature for autocatalytic systems, which is if you dope with the product, you will see increase in rates. So we moved ahead with this system since we can show ribozyme self-reproduction in these systems. So we went ahead to create networks in this system. So we did mutations in the IGS and tag pairings to create different kinds of ribozymes, and they are engineered in this way, like ribozyme 1, which is called UU, another ribosome is called AA. They are called by their mutations. And so we engineered certain networks such that ribozyme 1 can catalyze ribosome 2, ribosome 2 can catalyze ribosome 1, and similarly three-membered and more complex four-membered networks. And we did both of this analysis in the droplets and also the macrophage. And we do see, so here in y-axis, again, I have plotted the fraction of ribosome, intact ribosome that has formed starting from the fragments. And what we see is if only one ribosome like UU is there alone in any of these situations, so the UU does not really increase in concentration because UU can't self-catalyze by design. There is no self node like AU. AU has a self node. But the moment we add AA, or the moment UU is part of a larger network like AG and CA, three-membered network, or a four-membered network, which is a two-membered, three-membered, and four-membered network, we do see that UU starts forming. So that means not only UU is compartmentalized inside the compartment, then it would not form. So, but UU and AA, UU, and AG and CA, the other ribozymes are also in the compartment, as a result of which it's able to form. Similarly, we also plot the composition of both of them in the compartments, and this is the steady state compositional state. And similarly, we also did the analysis for the coacervate macrophage, and in the coacervate macrophage also we see a similar trend, though it's also interesting that in the coacervate macrophage, UU just by being alone also has can catalyze itself. So there seems to be certain background rate enhancement because of the condensed phase in the compartment, which is able to form UU. So, and on top of that, if UU ribosome is part of the network, yeah, it's able to form in much larger proportion. Then we went ahead to design a certain perturbation study. So here, so these are the networks that we designed, ribosome networks, and these are the nodes in which it can form. This panel shows the ribosome reaction in aqueous phase without any compartmentalization protocol, in which we see that, okay, yeah, and CA is added as a perturbation in the sense, so first we start with two ribozymes, UA and UG, and in aqueous phase without compartments, and we plot their distribution. We see this is their steady state distribution. There's more UA and less UG. Then we add a perturbation, which is designed to perturb the UG node, as a result of which the UG will now form more as compared to UA, and then the steady state composition shifts as compared to, so before perturbation, there's more UA and less UG, and after perturbation, there's more of the UG ribozyme and less of the UA ribosome. So this is what happens in aqueous phase. So the ribosome network gets perturbed and it produces a defined distribution. But then you wanted to study the same thing in compartment and macrophage, and what we find is if we start with two different compartments, one compartment that has network 1, another compartment that has only the node, which is used as a perturbation. The compartment kind of has a robustness towards the perturbation. That is, even after adding perturbation, even though UG is produced more as compared to the system in which there is no perturbation, still it's not flipped. In that sense, the order is still maintained. And uh, in the macrophase, which is the large compartments in which we started with UA and UG network inside the macrophase, then we added the perturbation from the outside, it seems that it kind of protects it much more. So in both of these compartmental systems, yeah, they kind of protect the compositional identity or the compositional distribution of the ribosome population as a function of compartmentalization to external perturbations, which is clearly not the case in the aqueous phase, in which any ribosome can perturb the network distribution. So this was the first part of the study. In the second part of the study, we wanted to introduce environmental fluctuations in the system. And we had few options, like to change concentration or to change the concentration of cofactors or to change the temperature. Went ahead with the temperature. And so again, it's the same protocol. At this time, we did a two-fragment study. It's only, we started with WXY plus Z and WXYZ. So it's a two-fragment study, and we only did it in droplets and not bulk. And the only difference is instead of incubating it at optimized temperature, we wanted to vary the temperature and see what's happening. And these are some gel images as a function of temperature this time. So this one is in bulk aqueous phase, where we see, okay, as a function of temperature, not much is really happening, though it's decreasing, as in the amount of intact ribosome that is being produced is decreasing as temperature is decreasing. But what is stark here is in the droplet phase, it seems that as you decrease the temperature, there is a certain temperature in which uh, the formation of the ribosome sharply drops. And this was interesting to us. So we went ahead to do a temperature scaling in which we studied the ribosome formation, the from two fragments in both bulk aqueous phase and in droplet phase. And these are some representative gel images. And what is interesting is at lower temperatures like 20 degrees, the ribosome formation still slightly goes on in the aqueous phase, the liquid phase, but it really does not go much, or it doesn't go on in the droplet phase. Where at higher temperatures, it appears to be enhanced in the droplet phase as compared to the liquid phase. So we uh, did the ribosome self-assembly under different temperature and quantified it, but we wanted to ask what is happening really to the ribosomes as it's in the droplet phase at varying temperature. So here you see, so this experiment is a fluorescence recovery after photobleaching, and here we did in two different temperature. The left-hand side is at 20 degree and the right-hand side is at 50 degree at higher temperature. And the fluorescence signal is again due to Alexa, which is being tagged to our ribosome and which is mostly localized in the compartments. And what we do is we create a region of interest where we bleach it, and in the beginning, and then we observe the recovery of the fluorescence signal, which is quantified here for 20 degrees and 50 degrees. So what we found that at 20 degree, the mobility of the ribosome molecules is quite less as compared to 50 degrees, because at 50 degrees, the fluorescence recovery is much higher, so they kind of mobilize themselves much more. But in 20 degrees, uh, the, the regions where the intensity was depleted kind of don't seem to be recovering that well, which is quantified here. So then we thought, okay, maybe because of because temperature affects mobility in phase-separated mobility of ribosomes in phase-separated droplets, and that's why it directly affects the self-assembly process. So we went ahead in characterizing the self-assembly of this ribosome for two different mutants at two different temperature, and this is what has been plotted here. These are two different mutants of ribosomes, and these are or this is in a droplet a coacervate phase, and this is in bulk phase. And here it's here the rates have been plotted as a function of temperature for easier visualization. and this is for another mutant ribozyme, again in the droplet phase and also the bulk aqueous phase. And here the rates are again plotted as a function of temperature for the droplet phase and the bulk phase. So what you find is different mutants of ribozymes also have a difference in the temperature scaling. At higher temperatures, the reaction rate is higher in the droplet phase, and in lower temperature, it seems to be higher in the liquid phase for certain ribozymes, but for certain other ribozymes, it's always higher in the compartment phase at any temperature as compared to
Nayan,
the aqueous phase.
Nayan,
Yes, yeah.
sorry to interrupt.
Yeah.
20 minutes are up. You might
I have
want to
one more
just
slide.
summarize.
Yeah.
Sure, sure.
Yeah. So now that we found the difference in the function of the ribozyme as a function of temperature, we wanted to have a selection protocol in which we take both the ribozymes in droplets and then we carry the system through temperature fluctuations between different temperatures like 20 degrees and 50 degrees, keeping the total time for experiment the same for total time of incubation same for all the experiments. So what we find is that as the like if we don't have any cycling and if both the ribozymes are at 20 degree, none of them really forms in the droplet phase. And if we also have no cycling between these two temperature and we keep it in 50 degrees and both the ribozymes form in the steady state distribution, which has been reported. But if we have cycling and if we have different cycling frequencies, we can manipulate the steady state distribution of the ribozymes, is that we can manipulate how much of one ribosome forms over the other ribosome. And there seems to be a certain cycling regime in which one can only have one ribosome as compared to the other ribosome in the system. So this is kind of a crude example of a selection in which one can have different, so one can have one ribosome outcompeting the other ribosome as starting from the same fragment pool. So with this, I'd like to end my talk and thank Karishma and Team India Bioscience and the expert mentors, Deepa and Abhiraj, and my supervisor Shashi, TCM members, Sudha, Priyan, Sandeep Krishna, and lab and the facilities that's there at NCBS. Thank you.
Great talk, Nayan. Thank you. Thanks a ton. Deepa and Viraj, go ahead.
Sorry.
Okay, maybe I'll go ahead quickly again.
Yeah.
Sorry, Nan, I'm a bit lost here. What is the connection of any of this to evolution? Can you remind me of that? Perhaps you said it and I missed it.
So I started by saying there's no connection to evolution. I applied in other. So I work in origin of life. And in that sense, they put me in evolution panel. So if you ask me if the connection, so I would say it's more connected to origin of life than evolution. Yeah. That's my short answer to it.
Okay. And then I have a question about this ribozyme work, the first part. Can
Okay.
you go scroll down a bit more?
Yeah.
So you showed these results where compartmentalization seems to increase the rate of the reaction completing.
Yes.
But there seemed to be a trade-off where the amount of final product that was being generated was not as high as when, here.
Yeah. So
What
that's
is going on there?
it's because of the carrying capacity of the compartments. So how much RNA they can, because if we start with some RNA in the aqueous phase and then undergo the compartmentalization protocol, there's a certain fraction of RNA that's always in the aqueous phase and the fraction of RNA that goes in so the aqueous phase is always like higher because it has more RNA. So its carrying capacity is higher in that sense. Yeah, so that's the answer.
I see. So is that not important for how these ribozymes will function and what you then infer and the implications for origin of life?
Yes, it is important. Yeah. So we have we did study it in this paper. So we have it in our supply figures where we changed the size of the compartments and we showed that and we also found that its maximum carrying capacity is at like 3 micromolar. Anything beyond that it doesn't take. So that's like a number. And one is to either change the nature of the compartment or increase the size of the compartment to increase the carrying capacity.
Okay, and I have a question for the second part where you showed that if you have multiple ribozymes in one compartment, that somehow increases the efficiency with which they self-reproduce. Could you explain to me what is the mechanism through which that's happening? Yes, here.
This one, no?
Yeah.
So it's basically Is my mouse visible? Yeah. So the thing is, so it's based on the network architecture. So the simple answer will be based on the network architecture. In that sense, so these ribozymes are called by their mutation. So I can call one UU, the other A, just for yeah. So UU cannot really uh, self-reproduce itself, starting from its fragments, but it requires an A because of the Watson-Crick pairing. So the mechanism is such that the internal guide sequence and the tag is mostly efficient because of Watson-Crick pairing, which is can be
I see.
catalyzed. U can be catalyzed by an A, and A can be catalyzed by U. In that sense, one can create a two-member network in which UU catalyzes AA and AA catalyzes UU, because that's how like AA has the complementary tag to the uh, IGS of
So
U.
it's not that, for example, in the first part, if you add UU and CC, that's not going to work because they can't.
UNCC is not going to work. So they may
Right.
form very little amount based on background reaction, but not in an autocatalytic self-reproducing manner.
I see. And
They may
so
just, yeah.
what is the connection between this and how you imagine ribozymes functioned in early life? I mean, okay, if you make complementarity, fine, they help each other and so on, right?
Yeah,
But
yeah.
it's not clear to me if that is thought to have existed early in life, or
So
is it a mechanism that you're proposing?
no, so it's not like, s- yeah, so there's quite a bit of discussions there actually. And currently people usually believe it's mostly the what's, so in a random pool of mutants, like in a random pool of RNA, and people believe it's mostly by evolving Watson-Crick pairing combined with certain Obel pairing to give secondary structures, one will get a diversity of functional ribozymes. But yeah, certain experiments have also been done in that. But yeah, in terms of dominant interactions, currently it's mostly Watson-Crick and Obel in the field. And yeah, so
I
yeah.
see. And finally, one just comment and suggestion that I think it would really help if you, like Pavitra did, if you pause in between and summarize what you're trying to tell us, right? Because you're showing a lot of data in your slides, and for people who are not in the field, it's going to be difficult to understand. So it helps if you summarize in between what's the main point, especially at the end. I really felt like would have helped me take some, takeaway messages that you want me to take away. Okay, that's it.
Thank you.
Thanks.
Thank you.
Thank you, Deepa. Thanks, Nayan. Viraj, on to you.
Yeah, thanks, Nayan. My point is, yeah, my major point was the same that Deepa raised at the end. That do you think you can right now summarize your talk in maybe 20 seconds to just tell
Yeah,
us that links
I
to
can.
what we started off?
Yeah. So I studied self-reproducing ribozymes, and I developed a protocol to compartmentalize self-reproducing ribozymes, and to show that compartmentalizing self-reproducing ribozymes can have certain benefits like enhancing rates, and also it can give robustness towards external perturbations from the environment to these compartments, and also under environmental conditions which are fluctuating, one can select for one ribozyme over the other ribozyme in a compartment. So that's mostly the three key takeaways.
Sure. All right. So a couple of other points I would mention, elaborate again on what Deepa said. I think it would be useful if each slide had like a take-home message or a summary at the end. That helps people hold on to that, and how what you do next really adds on to it.
I'll add it. Okay. Thank you.
And at the end, along with the summary, it would also be nice if you link it up to, how do ribozymes maybe help in comparison with proteins and DNA that you mentioned initially.
Okay, I'll take that.
Maybe help in giving us a broad view of maybe what was more likely.
Okay, got it. Yeah, I'll take. Yeah.
Yeah. That's it from my side. Yeah.
Thank you.
Excellent. Thank you, Deepa. Thank you, Viraj. Thank you, Nayan. And Nayan, yes, we did a little reshuffling to put you into this session because, we were trying to fit as many PhD researchers into as many sessions as possible. And we didn't get an equal number of applications across all, like molecular biology, cell biology. Deepa and Viraj got an overwhelmingly large number of applications. And then there were areas that got a fewer. So thanks, Nayan. Thanks for being on board for this reshuffling.
It's okay. Yeah. Thank you.
Yeah, thank you. All right. So we wrap it up. We had two speakers today. The feedback form is here for the audience, but for the speakers and mentors, it will go on email. But audience, please let us know what you thought of PhD Cafe overall. We've been receiving some really good feedback across the week. So we look forward to hearing from you too. And the giveaway winner is Pothina Amarnath. Thank you, Pothina, for keeping the chat window active. And email us at hello@indiabioscience.org to claim your prize. And yeah, that's exciting for you. Great. Um, Other than that, yes, welcome, Pothina. Other than that, thank you to mentors, speakers, audience, and Team India Bioscience. That's Arushi right there, Ankita, Manjula, Rutuja, and Shweta. Thank you all. This has truly been teamwork to get through the week. So thank you very much. And what we will do now is, I guess, get on with our work and our life till 1 p.m. when we will be back on another meeting, another Zoom link where Arushi will talk about transferable skills during a PhD and Ankita will chat with her about it. So see you at 1 p.m. and then we have an afternoon session. But for now, see you at 1 p.m. Thank you very much, all of you, for being here. Have a good day. Have a good rest of the day.
Thank
Bye.
you.
Thank you.
Thank you, Nayan.